This is part 2 of the recordings for chapter 9, and we are on slide number 48, and we're gonna learn about step 2 of the cellular respiration. So it says after pyruvate is oxidized, then you have the citric acid cycle, and that completes the energy yielding oxidation of organic molecules. What that means is, by the end of the citric acid cycle, glucose has been completely broken down, and all of the energy has been extracted from glucose that could be extracted. Okay, so so this is the step that it's the first part of step 2. I've always taught it as this is the prep reaction for the citric acid cycle. And what actually happens is the oxidation of pyruvate. Remember, there's 2 pyruvate molecules produced during glycolysis. So they're now going to be broken down into molecules called acetyl-CoA. CoA stands for coenzyme A. Acetyl is a chemical group, and it is attached to coenzyme A. So we just say acetyl-CoA. So pyruvate has 3 carbons. Acetyl has only 2 carbons. So that extra carbon is released as carbon dioxide. So during this part, oxidation of pyruvate, carbon dioxide is produced, more NADH is produced, and then we get pyruvate converted to acetyl-CoA. And that is going to enter, that acetyl-CoA is going to enter the citric acid cycle. But no ATP is produced during pyruvate oxidation. You don't need to watch that. The citric acid cycle, which is also called the Krebs cycle, well, I mean, I still wanted to see the inputs. Okay. All right, here we go. I wanted you to see how many of each. All right. Well, I don't see that it tells you, but all right, it's showing you here that this is what a molecule of pyruvate looks like. See, it has 3 carbons. Well, pyruvate is going to be broken down, and each pyruvate is going to release 1 carbon dioxide. It's going to produce 1 NADH and 1 acetyl-CoA. You see how the acetyl-CoA has 2 carbons now, and then it's attached to coenzyme A. So the thing is, you have to understand it's 2 pyruvates that enter, or that are oxidized, because 2 pyruvates were produced from glycolysis. So the 2 pyruvates would end up producing 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. And since that's not written anywhere, I'm just going to come back here to the notes and just say it. So for every pyruvate, and there are 2 that were formed in glycolysis, you're going to get a carbon dioxide, NADH, and acetyl-CoA. So really you're going to end up with 2 carbon dioxide molecules, 2 NADHs, and 2 acetyl-CoAs. And then the carbon dioxide is just given off into the atmosphere, and the NADH, as we've already learned, is going to be used in the last step, oxidative phosphorylation. But the acetyl-CoA is going to enter the citric acid cycle. So the citric acid cycle, which is also called the Krebs cycle, for the scientists that discovered it, is going to, it's a cycle or series of reactions, and it's called a cycle because it continues, the reactions continue as long as you have acetyl-CoA that enters into the cycle. But for every turn of the cycle, and there's 2 turns of the cycle because there's 2 acetyl-CoAs, you're going to get 1 ATP. So citric acid cycle total produces 2 ATP molecules because it turns twice. And you're going to get one NADH, but it'll end up being 2. That's wrong. 3 NADHs. The number 3, I don't see it in the notes. It's 3. Let's see if there's like a better... inputs and outputs, maybe. No, of course not. All right, so let's do it from the cycle. Let's talk about it from the cycle. Okay, here is the citric acid cycle illustrated as like a series of chemical reactions. Basically, it starts here. Acetyl-CoA enters the citric acid cycle by binding with a 4-carbon molecule called oxaloacetate. This would be step 1 of the citric acid cycle. Acetyl-CoA combines with oxaloacetate, forms citrate. Well, the reason it's called citric acid cycle is anytime you have a chemical that has ATE on the end, citrate, pyruvate, malate, fumarate, you can also say citric acid for citrate, or pyruvic acid for pyruvate, or lactic acid for lactate. Okay? Now, I do want you to know this first step. I want you to know the first step of the citric acid cycle is when acetyl-CoA combines with oxaloacetate to form citrate or citric acid. Citric acid has 6 carbons. Now, it may seem like we've just gone back to the beginning because glucose also had 6 carbons. But we're building this molecule, citric acid, because as it's broken down, we're going to produce some things. We're going to produce some products. So we're going to produce NADH here. We're going to produce NADH here. We're going to produce NADH here. That is 3 NADH molecules total. And then since there are 2 acetyl-CoAs that enter the citric acid cycle, you can multiply everything times 2. So it's going to be a total of 6 NADHs produced in the citric acid cycle. And then a total of 2 ATPs and a total of 2 FADH2s. And FADH2 is another coenzyme similar to NADH. Now, going on to the last step, during oxidative phosphorylation, you have 2 things. You have chemiosmosis and you have electron transport. And the main thing is you get a lot of ATP during this last step, because so far we've only made 4 ATPs. 2 were produced in glycolysis and 2 were produced in the citric acid cycle. So we still have, if we're going to end up with 32, we still have 28 more ATPs to be produced during this oxidative phosphorylation step. So one thing that you need to know is all of these accumulated coenzymes, NADH and FADH2, that have been accumulating in the previous steps, they are all going to enter the electron transport chain, and they're going to donate electrons to the electron transport chain, and that's going to provide the power. The transport of those electrons is going to provide the power to synthesize ATP through or via oxidative phosphorylation. I'm just looking through. I haven't looked through these yet. I'm sorry, but if I can find a picture where I can show you this, it'll be a whole lot easier. Yeah, great. This is a great picture. Okay, so this is what happens. And you can watch this. I mean, you can look at this picture. You can go back and watch the animation that I told you about in it's slide number 10. But I'm going to explain this picture to you because this shows the whole entire, everything that's important to know. Everything that's important to know about the last step, oxidative phosphorylation, you can actually see in this illustration here. So here's what we have. We have the mitochondria, And then this membrane, this phospholipid bilayer with proteins embedded, is not the plasma membrane. It is the inner membrane of the mitochondria. The inner membrane, because the mitochondria has an outer and an inner membrane. It's a double membrane organelle. So this is the inner membrane. And basically what happens is that the NADH molecules and the FADH2 molecules carry those hydrogen atoms. So they're going to carry electrons that are within those hydrogen atoms. So the electrons will be released from NADH and FADH2. Then those electrons are transported from carrier to carrier to carrier until finally they are added to oxygen and hydrogen ions, and water is formed. So the final electron acceptor is oxygen. That's where the electron's going to end up. It's going to combine with oxygen gas. And this is the role of oxygen gas, by the way. We haven't even talked about oxygen gas yet, and that was one of our reactants. So far, we've only been talking about glucose. This is where oxygen comes in, okay? Now, the oxygen is going to combine with hydrogen ions, which are also provided by the NADH and the FADH2. Because the hydrogen atom, when the hydrogen atom is released from the NADH and FADH2, hydrogen atoms carry electrons and they also carry hydrogen ions or protons. So the hydrogen ions are going to combine with the electron that just went through that electron transport chain, and then they're going to combine with oxygen and form water. So this is also where water is produced. All right. So basically we have electrons being transported. Well, the flow of electrons is electricity, which is a form of energy. So the energy from the flow of electrons down that electron transport chain is going to be used to pump hydrogen ions across the membrane. And then hydrogen ions are going to build up on the outside of this membrane. So they're going to encounter this molecule, and it's actually a transport protein. It doesn't look like it, but it is. It's a transport, a carrier protein, a channel protein. That was the word I was looking for. So when the hydrogen ions encounter this molecule, it's called ATP synthase, because it also not only is a channel protein, but it's also an enzyme. An enzyme that synthesizes ATP. So it's going to do that when the hydrogen ions flow from their high to their low concentration. They're going to flow across the membrane to the inside of the membrane. And the flow of those hydrogen ions is going to provide the energy to produce ATP. Now, another thing you can do, I told you that you could watch the animation on slide 10, but there's also an animation on slide 77 about the electron transport chain, and I highly encourage you to watch it. And another one, I don't like Bioflix as much, but this might, actually this was the one on slide 10. But I don't really like Bioflix as much as I like I don't know what this animation, what the source is of it, but I like this animation better. I can already tell looking at it. Bioflix sometimes can be a little bit complicated to watch, but go ahead and watch both of them and you'll learn something, you know. So during cellular respiration, so this is what we're going to do now. We're going to make an account of all the ATP that's produced in cellular respiration. We're going to list out all the ATP and which step produced it. So during cellular respiration, most energy flows in this sequence. It starts with glucose, and then NADH, electron transport chain, proton motive force, that's just talking about the hydrogen ions, and then ATP. That's the flow of energy in the correct order. Now, no process that produces energy, no process that gives off energy is 100% efficient. a typical gasoline engine, I think, like, I'm not talking about the newer energy efficient models. I'm talking about like an older, Ford or Chevrolet gasoline engine, is going to be about 25% efficient. So that's not good enough. And what that means is that only 25% of the energy from the burning of the gasoline is used to power the car. The other part is given off into the atmosphere as heat. So with cellular respiration, since it happens in all of these steps. Now, if it just happened as one chemical reaction, it wouldn't be as energy efficient. But because cellular respiration is broken up into all of these different steps, we get about 34% of the energy from the glucose molecule as ATP. And that ends up making about 32 ATP total. And the rest of that energy is lost as heat. So that would be the other, 66%. Okay, so we can look here at where all the ATP comes from. So we already talked about substrate level phosphorylation is what produced the 2 ATPs from glycolysis and the 2 ATPs from the citric acid cycle. But the NADHs that are produced in all the steps before the last step and the FADH2s, all of those go on to oxidative phosphorylation, and that's going to end up producing 26 to 28 ATPs, and it's going to end up adding up to 30 or 32 ATP molecules. This video is about the accounting of the ATP yield from cellular respiration, and this is slide 81. Good one to watch, I would say. Very good one to watch. All right. And then it talks to you about some chemistry and why it's not always something that it's not perfectly 32 ATPs each time. But I'm going to skip that. I'm going to read it real quick. Yeah, see, I used to teach that for every NADH, you got 3 ATPs, and for every FADH2, you got 2 ATPs. But what we've recently learned is you just can't be exact about what happens in the last step. So all we can say is that for different chemical reasons, you can get between 26 and 28 ATPs in the oxidative phosphorylation step. Okay, so now we're going to learn about fermentation. Let me see which slide. Yeah, this is a good stopping point. I am going to stop here, and the next recording I will start with slide 85.